Yeah, so I'm Katie. It's great to be here with you all today. I will try to make this short. Feel free to ask me more details about any pieces of my story later. So I went to a small liberal arts college in Maryland. I studied chemistry, biochemistry, and then came to Yale for my PhD in 2006. And I was in the chemistry department there. I definitely wanted to lean more towards structural biology and biochemistry. So I was in the biophysical chemistry track, and I joined a lab in MD&B. So I joined Scott Strobel's lab, who is now on the West Campus, but at the time was on Science Hill. And so I did a lot of X-ray crystallography on novel structured RNAs that were identified in Ron Breaker's group, and did some biochemistry and biophysics as well. And then, so that was I started in 2006. And then in 2011, I graduated. And then right when I was finishing up, Scott, my PI, was tasked with taking on the development of the West Campus, which at the time there was not a whole lot there. And so I agreed to stay on in his lab for a few more years while he kind of took on that greater administrative responsibility. And so I don't know exactly what my job was, but I kind of ran the day-to-day operation of his lab. So I still did my own research. So I was sort of like a postdoc, but then I did basically anything else that needed to happen. So helped mentor students and postdocs. I did like all the budgeting and kind of overseeing finances on the sort of day-to-day, week-to-week, month-to-month level. I took meetings that Scott couldn't take. I helped write grants and grant updates, and dealt with, basically just whatever came up. And towards the end of that, or towards maybe a few years in, I kind of was like, I feel like I've learned so much through this, but I really know this isn't where I want to be long term. And so, to make a very long story short, I ended up getting interested in the startup community, and I actually just kicked myself out of Scott's lab because it was a really comfortable place to be, and I knew I would just keep staying unless I kind of took the plunge. So I quit without knowing what I was going to do next, which not for everyone, but that's what I did. I started kind of going to a bunch of events in the startup community around Yale and the greater New Haven area. Got connected with people at OCR, and applying for a job at Arvinis, which at the time was a few years old, was maybe 30 people. And try to tighten up this part. Long story short, again, so Craig Cruz, who was one of the founders of Arvinis, saw my resume through the process and thought my background might be a good fit for a new venture he was starting with Keenan Partners. So I ended up actually being the first employee hired for that new venture. So got to kind of see company building from the very ground up. We ended up not continuing after several years. Just we made good progress, but it didn't seem like there was enough to kind of go forward and raise the next round of money. And so then also got to see the process of shutting down a company. And at that point, Arvinis had an opening that was a good fit for my skill set. So I moved over there. I'm a project leader, so I lead one of the oncology programs. And so, we can get more into what that looks like day to day, but I lead a team of scientists in drug discovery. Okay, that's probably too long, but that's me.
No, thank you, Katie. That was perfect. Matt, do you want to go next?
Sure. Thanks, Molly. So thank you also, to both of you for the invitation for being here today, and looking forward to the chat. I learned a lot more about you, Katie, I think already, just from your little blurb.
Thanks.
So I'm a Connecticut native, actually, and so grew up in Connecticut and have lived here most of my life. I went to the University of Connecticut as an undergrad, and really focused on on molecular biology and biochemistry. I started my PhD at Yale in 2003 and was just down the hall from the Strobel lab. I was in Andrew Maranker's group. And in Andrew's lab, I actually got a chance to do structural biology, which is really a primary interest of mine. So X-ray crystallography, as well as pretty broad protein biophysics. And I had a great experience there. I finished up my PhD in 2008 and then moved to Memphis to do a postdoc at St. Jude Children's Hospital with Brenda Schulman. And so in my postdoc, I continued structural biology, but I switched over really to the ubiquitin proteasome system and was understanding E3 ligases. And I did a postdoc for about 3 years. And then from there, I moved directly to Pfizer. So I've been at a pretty large company the whole time. It's almost 10 years, which is hard to believe. And I started in the department that I'm in now, actually, in the structural and molecular sciences department, originally as a senior scientist, and then went through a few different roles in the same department. So became a principal scientist and then a senior principal scientist, all as sort of an individual contributor. And then I took over as a lab head for essentially a crystallography group and served in that role for a few years. And then last year, I took on the lead for the department as director for SMS. And really what we do is support early stage preclinical discovery for inflammation, internal medicine, and rare diseases for Pfizer. A little bit of oncology work and a little bit of vaccine work, but most of what we do is to support Pfizer's small molecule portfolio. So we have a really great broad team of scientists that cover NMR and protein science and molecular biology, cryo-EM, X-ray crystallography, and we really work closely with chemistry teams to try to advance small molecule therapies, really from conception to clinical candidates. And that's where I am. So happy to chat more and answer questions later. But that's it in a nutshell.
Thanks, Matt. Sam, you want to head up?
Sure. So thanks so much for allowing me to be here as the only non-affiliated Yale person here. I'll be giving a webinar in very near future to you about drug development. And during our introductions at that time, they found that my journey is a little interesting, so that's why they invited me to meet with you guys. So I'm originally a Canadian citizen, and I got my honors biology degree in Canada, in Southern Ontario. And then I did not know what I wanted to do. So I applied to a couple grad schools and got into the University of Tennessee, Memphis. So where Matt postdoced, and basically got my PhD in molecular biology, learning protein chemistry, understanding gene transcription of the human apolipoprotein B gene, and then went and joined an immunology lab for my postdoc in Texas. So moving further south away from Canada, really learned a lot about NK cells, signaling pathways, and then had a chance to become faculty there for a few years before taking the opportunity to return to home to Toronto, Canada, where I joined a small biotech company. At that time, it was just emerging out of Queen's University and was about to relocate to the Toronto area. So we only had about 10, 12 people in the company. And I was there for 4 years, where I was initially brought on as a bench scientist because of my immunology skills. I took over their lead program, revamped the screening paradigms for their lead candidate for mild to moderate psoriasis, and learned toxicology as we moved through the drug development process, taking that lead compound to phase 2 clinical trials, as well as accepting other programs in autoimmune disease and oncology. Then I had the opportunity to join Charles River in the Reno, Nevada site. So moving from Canada out to the West Coast. And I joined them mainly because they had NHP experience. And so that is a premier site worldwide to conduct monkey studies. And I had, by that time, small and dog toxicology experience, and this was a chance to expand my repertoire. And that was 14 years ago, and I didn't think that I'd be staying with Charles River that long. And at that time, they were growing by acquisition. I was there for almost 9 years before they had reopened their Shrewsbury site in Massachusetts, where I'm close to right now. And so I came out there to become a director of toxicology, but I really missed drug development and opportunities where I could have left Charles River and joined certain biotech companies. They always enticed me to kind of stay in. And so then I joined the current group that I'm right now, which is Scientific Advisory Services Group, and was promoted a couple of years ago to director. And we're a collection of scientists that have extensive drug development experience. And from my travels and from my experience with Charles River, I'm able to work with clients in developing research strategies as early as drug discovery and target validation, all the way through the drug development process to pre-approval from a non-clinical and regulatory perspective. I work on a wide range of drug modalities, including small molecule, large molecule, oligonucleotides, advanced cell and gene therapies, and vaccines, as well as excipients developments. So a wide array of experiences. So I consider myself more of a generalist than a specialist in this realm. So thank you so much, and look forward to sharing more about how I got to where I am, some of it by luck, some of it by strategy.
Awesome. Thank you so much for that introduction, Sam. So I'm going to ask one or two questions, and as our panelists are answering, you can feel free to think of other questions that you have for them, and you can put them in the chat where Harini will kind of collect the questions that we get and pose them in the future. So my first question for you guys is if you can take us through what a week in your current role is like. You know, what do you do on a day-to-day basis? How do you spend your time at the bench versus in meetings versus other activities? So Sam, do you want to start this one?
Sure. So in my current role, I work with clients across the globe. So I'm not regionally defined, even though most of my clients are in the Cambridge area, as well as New York, Philadelphia, and in Washington, D.C. and Maryland. And so I mainly work from home remotely. That was pre-COVID. Most of my times are spent on teleconference calls, video calls. I would say about 40% of my time are directly with interactions, both internally and externally. And the other times I'd be more working on developing these strategies, reviewing regulatory guidances. And pre-COVID, I'd be traveling. I'd be going to these areas as well as sometimes to the West Coast to meet with clients, defining and supporting our business development colleagues. And so I'd be traveling at least probably about once a month out of town, and then into Cambridge about 2 or 3 times a week.
Katie or Matt, one of you is welcome to answer.
Okay, I'll go next. Yeah, so my like a typical week involves a lot of meetings, a lot of interacting with people. So I have like my team that I lead one-on-one with each of them each week, as well as run sort of cross-functional meetings. So right, we have kind of our biology group, but then I have a chemistry counterpart. We work with like we have an internal in vivo team. We have a modeling group, a structural biology group, and all of those teams we work with to discover and develop drugs against the target that I'm leading. So it's a lot of like coordinating amongst different folks, getting everybody on the same page, kind of defining the strategy of how we're attacking this target, and then making sure it gets executed. I make a ton of presentations. I feel like, right? That's a big part of my role, is just keeping people in the company updated on our progress. I am just starting, like I'm giving a talk at a conference later this week. So that's something that will happen occasionally. Yeah, I think mostly that. I am not at the bench anymore, so I have been working entirely remote since March, but my team is all on site. So Zoom is my constant companion these days.
All right, and I'll briefly chime in. I think my my current office 4 days a week is what you see in my background, which is my basement, sadly, which is both good and bad. That is purely a result of COVID. So I'm on site about 1 day a week at present, but prior to COVID was on site all the time. And sadly, I'm not at the bench at all anymore. So I was originally at the bench, 100%, and then that gradually changed. And so I'm not a bench scientist at all. Similar to Sam and Katie, I do spend quite a bit of my time in meetings. I think the good part is, even though I don't get to physically generate any of the exciting data, I do still get to talk about science and think about science quite a bit. So even though there are some of the activities that Katie talked about earlier, like budgeting and some HR issues and things like this, there is still a lot of time that's spent talking about science. I think the biggest part of my job now is really decision making, I would say. So it's a decent sized group. There's about 35 members of my department, and we oversee somewhere in the order of 35 programs. And the big challenge all the time is really to make sure that you're making the most efficient use of resource, that you're working on the key questions or challenges for each target, that you're generating decision-making data, and that you often have to make calls with only part of the answer. So I think a lot of time is spent on making calls on what to do next, and moving forward on exciting projects. Sometimes having to pause on less exciting ones, but trying to do that in an efficient and exciting way, to keep a portfolio moving. So I think a lot of my day is sitting in this chair, having discussions like this, sometimes looking at great data, and sometimes a bit more boring than that.
Katie and Matt, you guys sound almost a bit wistful about bench work, like you miss it a little bit.
Yeah, for sure. I started off as a bench scientist, and I do miss it. I mean, I think that having been in academics, biotech, and now the CRO world, every different arena has its specialty. There are things that are better than any of the others, but then there are things that you really don't really care for amongst the others. And I think that you have to kind of balance it to what will make you most happy.
Good advice. So my next question is, if you think about, let's say, one important transition in your career, because some of you have had many transitions in your career so far. What were your considerations when you were making this transition and looking for your next role? Were you thinking about the size of the company, what they were researching, what the company culture seemed like? Like what was most important to you at that point, and why? Well, I guess we can start off with Matt this time.
Sure. Maybe I'll try to be brief, but I'll mention two, I guess. One is maybe the transition from postdoc to career in industry. And I think I didn't come out really deciding that I wanted to work for Pfizer. I just knew that there were certain things that I really enjoyed doing. There were certain things in science that excited me. And I think I was really open to sort of where those opportunities would be. And I can remember, even considering academia or industry, and even talking about it with my my grad school advisor, Andrew, at the time. And I think I just tried to look for opportunities that were exciting. So I think I tried to keep a really open mind, not necessarily think that I knew exactly what I wanted to do either, recognizing that things change and careers evolve. But I think, I applied for, a collection of different positions. Some of them, were a little bit different. They weren't all quite in structural biology. They weren't all at big companies. But I did make a choice ultimately on a place that felt like it was a good fit. I was going to have an opportunity to do good work. It was a great team that was really productive and people were happy. And that really, long term is very difficult to predict anyway. So I think I tried to make a call early on that seemed like it would put me in a good spot, from the start. The other transition, I guess, to briefly mention that I think is probably a significant one is when you go from being sort of an individual scientist to being a people manager, and then suddenly having colleagues report to you. And I think that's something that, we don't really get trained for as scientists typically. And sometimes the transition can be really abrupt and it's a whole sort of different set of responsibilities and challenges. And I think I've really, really enjoyed it, working sort of with a team and having sort of individuals report, but I think it is a really, really different experience. And I think, there are parts of it that are appealing or not appealing to some individuals. And I think that, at Pfizer, you can do, you can advance in your professional career without supervising individuals, right? So you can be an individual contributor or a people manager. You can go M track or research track. There are different ways. You don't have to be a supervisor to advance, but it does present different sets of opportunities, challenges, and sort of satisfaction. But I think that is a big jump, a transition at least.
Katie, do you want to go next?
Sure. Yeah. So, I think when I was making the transition from, working in an academic setting to, wanting to check out biotech or, kind of startup, I think for me, I wanted to, I was looking for something where I could help build something from the ground up. I know that's something that I've always really love doing, coming in where there's kind of like a blank slate and saying, what can we build here? And I think I also, for that first role, I think I probably didn't even fully know how much I didn't know, but I appreciated at least enough to know that I knew nothing about company building or what it looked like to, discover a drug and take it through development. Still, obviously, I'm at the early end of that, so learning a ton. But I think I wanted, my dream, and I feel like I got to have this in so many ways, which I'm so grateful for. It was something where I could see a lot of different pieces of it, and get exposed to a lot of different things. So I think I didn't know what I wanted. And so, joining a really, really early stage startup, I got to know the VCs, so venture capital people. I worked really closely, that was probably the person I worked the most closely with day to day was somebody from Canaan Partners, so a VC. So I got to kind of see that piece of it and oh, what would that be like? And what is their role like? And how does the money work? And what's seed financing? And what's a Series A and a Series B? How does all of this work? And I got to have, kind of experiences in hiring people. And, like Matt said, we don't really get trained for that at all, but it's just sort of trial by fire. Okay, well, let's go hire some people. Okay, let's write a job description. Let's interview. Let's check references, and you're just calling people up and trying to pretend like you know what you're doing, asking. And, right, doing everything from, this was not the exciting part, but, talking to Comcast and getting phone lines set up and ordering lab equipment. And, and I think through that process, really, this is maybe the thing I'll end with, and I was at the bench a ton, right, in that role, along with doing everything else. And I think for me, I found, even if I maybe sounded wistful about the bench earlier. I really found, like, for me, that I would be happy moving away from that, and I loved more like the people aspect and the decision making and strategy and right, looking at data and helping other people, but not being the one that actually was doing it. And so when I moved to Arvinis, that was a real big driver for me, that was the type of position. And also the person who I would end up being my boss, I had gotten to know him a little bit, just through like a mentoring thing. And I felt like he was someone I could really learn from. So I guess for me, the theme at each stage was like really feeling like I could learn a lot in the next role. I think that's the key thing for me.
Yeah, I think my answer is going to be very similar to both Katie and Matt. I think that transitions can be hard when you're moving from one industry to another and not really knowing what those are. But I think that looking for opportunities that match your personality really help. For me, when I was looking for a new opportunity after my stint with the Canadian biotech company, I had close to 10 offers, and I was surprised that I didn't take a job in Boston with a large biotech company. I actually took it with Charles River, who I'm with today, Reno. Little did I know that I'd be moving basically back to Boston, because that's where I'm situated today. And I think that the key thing about it was that when I made that decision to choose the CRO world, was kind of like what Katie said. It was an opportunity for me to learn more on something that I didn't have, and that really excited me. And the other aspect of it was, at least with the large pharma that I could have joined in Boston, the people, the group that was very formal, very different for me, and I just didn't feel like a good fit there. They'd be like, it felt like a job. Whereas going out to the West Coast, it was amazing. It was just like night and day difference. And the people just they were all transplants. So that kind of helped me out too, is that, you almost had a family there. So to me, the working conditions, the interactions with people made a major influence for me to join Charles River. And I only had joined Charles River for potentially 3 years, and 14 years later, I'm still with them. And so you never say never, and always keep your options open. And even when Charles River, one of the reasons why I'm still with them for such a long time, it's kind of like what Matt did. Within Pfizer, he had many different positions, and I think it's probably because he just said, yes, that sounds interesting, I want to learn more. And that's what I did within Charles River. I just kept on moving from just simply a GLP study director to program management, to client portfolio management, to director of talks, then becoming almost like an internal consultant for our clients, what I'm doing today. And it's just being open. And, even though I'm away from the bench and I do miss the literal science of it, all my experiences in the past have really I can actually use elements of things that I've learned way back when to how I evaluate data in my current role. So everything is kind of cumulative, and you can leverage it.
Okay, I think we're going to stop the recording now.